you how to set up Punnett squares so you can figure out the predicted genetic and phenotypic ratios of the resulting offspring from certain crosses. The book does go over this, but some of the labs don't really explain this too well. So I'm going to go over just a couple problems that will hopefully help you figure this out a little better. So you want to try to figure out what possible genes each of the parents have, because every female has 2 copies of every gene and every male has 2 copies of every gene. And when they make their gametes, each gamete gets one of the possible genes. So you do that, and then you figure out what the possible gametes someone could make it. So let's just hop in and with a couple of these, and you can see how it works. So let's just say you have someone with freckles, which is dominant, but they are carrying the non-freckle gene, and you want to cross that person with another person who has freckles, and they're carrying the non-freckle genes. So first of all, whatever the dominant trait is, and in this case it's freckles, generally the capital letter from that is used for the dominant trait. So capital F means freckles. No freckles is not N, it's just lowercase f, meaning it's the recessive to this dominant freckles trait that is being displayed in both of these parents. So the first thing you want to do is figure out what the possible gametes that each of these parents could make if they were to go on and combine them and have a baby. So this parent has this gene, because we know when you go through meiosis, half of them will get this gene and half of them will get that gene. So you will have some of your gametes will have the dominant trait and some of them, and it'll be a 50-50, because every cell in this adult has 2 copies, 1 dominant, 1 recessive, and when you split that, you're going to get a 50-50 mix of gametes having 1 trait for dominant and 1 trait for recessive. And because they're going to combine with another human, they only want half the number of traits to come from the father, and the other half will come from the mother. And that's why during meiosis, instead of passing on both of these, you just pick one, and then the other one comes from the other parent. So this parent also will have a 50-50 ratio of dominant freckled gametes, and then gametes that carry the allele for no freckles. So what you do is you set up your square. I'm sorry, I borrowed this whiteboard for my friend's daughter, and it had said Franklin's Room Forever, and this will not come off. It was on here so long. But then you put at the top the gametes from the possible, well, not the possible, we know the parents, the possible gametes from the definite parents. So this parent will have this gamete and this gamete possible, and then this parent will also have the freckle gene and then a non-freckle possible. And then from there, you just carry these over, so capital F, recessive non-freckle, and then you bring these down from the top, so capital freckle, little f, and now you have the possible genetic probability of the offspring, basically. So you count up which is which, and then that'll give you the ratio for the genotype. So for this, the genes, it's telling you right here exactly what the genes are. The phenotype will be whatever trait they're showing. Phenotypically, both these parents were phenotypically they're both freckles. That's all we could see. But genotypically, they're carrying a recessive allele, both of them. It's just not being displayed. So the genotypic ratio for this is, we have one that has both dominant alleles, so homozygous dominant, one of those, and this one dominant, one recessive, one dominant, one recessive, They're the same. It's just when you brought it down, this F is first. But they're both heterozygous, just like this parent. So there's two of those. So two heterozygotes. And then you have one homozygous recessive, which is no freckles. And if it asks you for the phenotypic ratio, so everybody who has at least one of the dominant capital F alleles is going to be showing freckles. So these three, which is this one and these two right here, are all the same phenotypically. So you're going to have three with freckles, and you're going to only have one that is no freckles. And the phenotypic ratio, those are words that describe something. Like freckles, no freckles. Genotypes are two alleles, and you have to list which each allele is. So that is how, if you had this problem and you needed to figure out what the genotypic and phenotypic ratio for a heterozygote freckled parent with another one, this is what you'd get. 1 to 2 to 1, homozygous dominant, 2 heterozygous, and 1 homozygous recessive. And then 3 freckles for 1 that doesn't have freckles. And this doesn't guarantee what the offspring will look like. It's like the probability for flipping a coin. It's 50-50 when you flip it. It doesn't mean you'll always get that. And this is just saying the probability that you're going to have a freckled child, these 2 freckled people who are both hiding the recessive allele, is they'll have about 3 freckled kids for every 1 non-freckled kid. And that's just the expected probability. It doesn't dictate, you know, there's those people that have like 7 boys in a row and, you know, it's a 50-50 boy versus girl, but yet, you know, you just keep having whatever. So let's go over another sort of problem. We can get this off. I tried to kind of oil it before I put my writing on top of Welcome to Franklin's Room. So the next type of thing we're going to look at is, what if you have a disease where it's dominant? So if you receive the gene at all, you express that trait. And such a disease is Huntington's disease. The allele isn't super common out there, but if you receive even one of the dominant alleles for Huntington's, which is H, regardless of what's here, you will have the disease. So in this situation, let's say if someone who's or heterozygous for Huntington's disease, which means they will eventually display it, is crossed with someone who is homozygous recessive, doesn't carry the allele for that disease at all, then what will that cross look like? Okay, this parent, they can make alleles with the Huntington dominant gene, and then half of their gametes are going to have the allele that has the recessive trait, which is not having Huntington's. And then this person, they can make this a small h, or they can make this small h, which is the same. So basically, they only have one possible gamete, and it's going to be, these are their eggs and sperm. It all 100% of this person's gametes are going to have this allele because they only have that allele. There's no other possibility for them. So what you do here is you make out your square again. Put the possible gametes down the side. Huntington trait with non-Huntington. That's your heterozygote affected. And then they're being crossed with someone who is non-affected, which is homozygous recessive. And then you just bring your H across again. Little h, and then these are all little h's because that's all they had there. So if you wanted to know what's the probability of someone with Huntington's disease, that's a heterozygote having an offspring with that. Well, how many of these in the square have at least one H? And it's 2 out of 4. So you're going to have 2. That doesn't go there. Sorry. 2 heterozygotes affected for every 2 homozygous recessive non-Huntington's carrying children. So this is your genotypic, because it's described this way. And if they ask for the phenotypic, it would be 2 affected to 2 non-affected, or just not Huntington's. But in that case, it's a 1-to-1, because this parent only has one possible gene they could have. And then there's another sort of situation. Generally, so far what we looked at, there's straight dominant and recessive, where if you get the dominant gene, that's what you show. The only way you're going to show this recessive is if there is 2 of those. So what we're going to do now is we're going to talk about blood types, where there's actually 2 different versions of a dominant allele, and then there's O. And those deal with what sort of sugar chain is on the outside of your blood cells. So you can have A or B, and they are codominant. Or you can have O. So the only way that you people are O blood type is that they did not receive an A or a B from either parent, and they only have the O allele. So in this case, let's talk about, what if you have a female who's AB, which is kind of rare, and she marries a male who is A blood type, but he's carrying the O allele. So if you're AO, your blood type is considered A. Same thing if you're BO, your blood type is B. And if your alleles are AB, your blood type is AB. It's called A. You describe them both as that. So we're going to do the same thing. I'll draw the little male thing. Okay. So these people got together. They want to know what the possible blood types are for their children, which is a good way to trace who the parents of these kids really are if you're questioning. So let's do the square again. This one only is going to have half with the A and half with the B. So A is their gamete and B is the other half of the gametes they'll make. This individual, half their gametes will have A and half of them will be the O gamete. And then we just carry these over, cross, B, B, bring down the A, bring down the O. So the genotypic ratio of these children, because we have 2 codominant blood types, there's 4 different genotypes possible here. Genotypically, you have 1 AA, which is homozygous dominant for A. So their blood type will be A, to 1 AO, and their blood type would also be A, to 1 that is BA, or AB is how they usually say it. No one's ever described their blood as I have BA. My blood type is AB, which basically means they have the A and the B. They just always say the A first. It's the same thing. And this is BO, which is blood type B. So that's the genotype typic ratio on that. And the phenotypic for that, would be, these 2 are both A, because even though they're carrying the O, A is dominant. If you have any of the A chain in your blood, you basically cannot get blood from someone who doesn't carry this, because your body will attack it or whatever. So these are both considered A. So you have 2 A bloods to 1 AB, again, not BA, 1 AB blood type to 1 just B. You don't describe as a phenotype, those are the recessive, so you just say the dominant. So this would be the blood type breakdown for these 2 people who have very different blood types going in, and it gives a whole weird range of blood types. B is very rare though in people. Almost everyone is A, O, O. I used to do blood types as a plasma center, and There'd be hundreds of people who were A and O to maybe 1B, and then several hundred more that were A and O. So B is kind of rare, but that's how this would work out. And I'm going to do one last one for you because I know this is getting boring, but I do just want you to see how these all will work out. And the last one I'm going to do is where you know the phenotype of someone, but you don't know their genotype. So what you can do is called a test cross. Like if you crossed brown dogs with white dogs and brown was dominant, and you had a random brown puppy and you're like, well, what's the genotype? Brown is dominant. This is brown, or capital B. And little b is white, recessive. I was going to try to get some dogs to help with this, but I don't really have time for that. And they're not that good at helping. They are cute though. So if you had a random brown dog and you're trying to find out if it's carrying a gene for white fur, what you can do is cross it to the recessive phenotype, because that way you know that phenotype is homozygous recessive. You know it only has, so you have a B question mark crossed with somebody who's homozygous recessive for the brown or white. And then you look at what the offspring look like. So the question here would be, what is this individual's actual genotype? We know they have one dominant gene. So you fill this in with a question mark. It's B, question mark, B, B, carry this big B down, carry this question mark over, and then fill in little b with everything else. So here's how you figure out what this question mark is. You look at the offspring, and if 50% have the white again, which means it would have to be he was carrying the B, if that's what this was secretly, then you would get a square that works out where half of the offspring are brown because they contain one of these, but the other half only have the recessive gene, so they'll be white. So if you see half and half, you know that they were carrying the recessive gene. However, if this individual was homozygous dominant and this was another dominant gene, these would be dominant alleles, and basically every single possible offspring would have the brown fur. So really, you just breed to the homozygous recessive, and if you see any recessive offspring, then you know this individual that you were questioning was carrying the recessive gene. Thank you for watching this. This is very important, I think, to watch before you take the next test. So good luck. I feel like I'm taller than where the thing is in my head was probably cut off. Sorry.